Hello everyone. Today I, Shipra Banerjee, Associate Professor of Brainware University, will be presenting the topic on biochemistry and clinical pathology. So there are different topics. First of all, we should know what is biochemistry. The chemistry related to the biological of human origin. So there are different macromolecules or macromolecules present in our body. So one of the most important compound in our body is carbohydrate. Okay? So here we'll be discussing about the carbohydrates. So you know very well carbohydrate is the basic food which is required for the energy production in our body, and it is composed of carbon, nitrogen and oxygen. Sorry, not nitrogen, carbon, nitrogen and oxygen. And the simplest moiety is the glucose. Okay. So one of the now I'm elaborating the slides. Carbohydrates, one of the nature's three classes of organic compound, and the two being fats and proteins. You know very well. So this is composed of carbon, hydrogen, and oxygen. So there may be three classes. That is the monosaccharide, oligosaccharide, and polysaccharide. So glucose moiety is not a simple compound. They may be of single heterocyclic. There may be two heterocyclic units, more than two, and polysaccharides. Poly units may be. So monosaccharide is a single moiety glucose unit, so which cannot be further hydrolyzed. If we want to break down the structure, there will be carbon dioxide and water molecule. And this is the building block of complex carbohydrates, cellulose, starch. These will be composed of carbon. Now coming to the oligosaccharides, that is the low molecular weight polymers that is composed of 2 to 3 or 4 or 5 monosaccharides are composed. So whenever two monosaccharides will be attaching each other, they will be as a disaccharide. And trisaccharides, 3 monosaccharides or 3 glucose will be attached. There may be 10 glucose units can be added to them. Okay. So now coming to the polysaccharide. Now polysaccharide is the most important compound, which is the complex carbohydrate, and it is found in the starch and glucose. Now coming to the elaborating the structures of monosaccharide. So here, if 3 carbon is present, that can be known as a triose, and it is known as a C3H6 group. Dihydroxyacid and glyceraldehyde. Basically, dihydroxyacid and glyceraldehyde, they would be not known as the basic carbohydrate or glucose moiety. Whenever any glucose moiety will be broken down, they will be forming this triose. Now there will be tetrose, erythrose and triose, pentose, arabinose, deoxyribose, ribose, and xylose. These are the important some carbohydrate. Now the DNA, where deoxyribose is a pentose carbohydrate that is present in the DNA molecules. Now hexose, that is the basic and the conventional glucose, that is galactose, glucose, and fructose and maltose. Now coming to the oligosaccharide, here two disaccharides are there. One is lactose, maltose, sucrose. This is the different carbohydrate present in different compound. Lactose can be found in milk. Maltose can be found in different form, different starch or different grain. And sucrose you can find it in sugar or in different fruits that produces the sweetening taste. Okay. Now coming to the trisaccharide, that is raffinose, that is also present in some of the fruits and fibers. Okay. Next, tetrasaccharide, stachyose, that is one of the carbohydrate present in different product like this. Okay. Now, there are some polysaccharides, pentosane, araban, and xylem, hexosan, cellulose, galactan, glycosan, and starch. These are the different hexosan, which is a very complex carbohydrate. Okay. And complex polysaccharides are like gums, hemicellulose, and pectins. That is also present in some plant in case of the scale of the plant and the bark. Here you can find this pectin or hemicellulose or gums. That is a modified glucose units also. Okay. Next, coming to the polysaccharide, that is more than 10 monosaccharide units are joined together. They can be hydrolyzed into 100 or even 1000 on monosaccharide units. Like whenever you are just digesting any rice or starch. So the glucose molecule will be liberated to small groups. Okay. So suffix -ose in sugar, that is changed to ase, that is polysaccharide to different carbohydrate molecule will be broken. So example pentosans. So pentosans to pentose. Okay, they will be converted to pentose. Xylan, paravans, hexosans, galactan, mannans. These are the different polysaccharides. Glucan, like starch, dextrin, glycogen, like starch, dextrose can be found in corn, like mutta, maize. Okay, different compound, different starch. Like you can found the oats is very good complex carbohydrate that is used by different young generation for the weight reduction. Okay, rajma, sorry, not rajma. You can find ragi and say um, that is dalia. There are different type of carbohydrates. Then GAMS, then mucilase, then algal polysaccharide, acid, and carrageenan. Complex carbohydrate which is used by the space researcher. Okay, bacterial polysaccharides, xanthocam, pectin or pectin substances, another different type of polysaccharides are available. Classification of polysaccharide, different structural types are there, infinite variety. They are broadly classified as homopolysaccharide and heteropolysaccharide. Homo means you know very well the all the units of the polysaccharides are same. Like in case of starch, if you take the starch, there will be amylose and amylopectin. And in case of heteropolysaccharides, so different type of saccharide will be there produce a complex polysaccharide. Now, homopolysaccharide coming either simple linear structure or branch structure, varying complexity with one type of that unit link. That I have said the same structural unit will be present throughout this structure, like fructan, glucan. So here starch and glycogen will be present. Okay. Next, we are going to the next slide. Perfectly linear polysaccharide, they are single neutral monosaccharides structurally only one type of species of water, can be solubilized only under drastic condition. Also have the tendency to precipitate from solution. Retro Next, branch polysaccharide, there will be some change in interaction are less pronounced in linear polysaccharide. Some you can find some of the polysaccharide you can form, okay, and some polysaccharide like corn flour. Okay. Next, sabudana flour, dessert, wheat flour, different flours are there. You can find the consistency is different. Here, that means uh, structural alignment and linear alignment are different. Okay. That's why their consistency is different. Solubility is also different, more soluble in water. Solution of lowest viscosity and lower tendency to precipitate. Okay. Suppose the polysaccharide is a branch having four branches. So having will be whenever they will be solubilized, they will be forming a viscous solution. So that's why their viscosity will be very low and lower tendency to precipitate. And they will not be precipitated. Okay. Suppose you make a starch solution, a rice starch solution. They will be having lot and they will be making colloidal solution from sticky paste at higher concentration. So if you make too much concentrated solution, then they will make a sticky paste due to side chain and side chain infection. Suitable as binder or is where it can be used. This type of binder, plate starch and starch can be used. The tablet binders and medicine preparation it is also used. Okay. And different food preparation is also used. Okay. And this can be found in our body also in like Maggi, pasta. Here the different polysaccharides are used, which is not good for our health also. It's one of the point that's why you should study it. Now coming to the linear branch polysaccharide, what are the properties? The combination of perfect linear and branch polymers, long backbone chains with short side chain, which is alkyl cellulose, and long backbone chain with high viscosity of the solution. That means we are just describing the problem. Numerous short side chains weakens the interaction between the molecules, and if there is numerous side chain for different compounds, that will weaken the interaction between molecules. As a result, there will be no interaction, so chains will spreading here and there. Good solubility and rehydration rates, stable and highly concentrated solution. There will be not so much hydrated, and we can use for rehydration. There will be stability of the. Now, modified polysaccharides, like physical and chemical methods can modify its properties of polysaccharide products of suitable for specific purposes, for making different medicine. Binding of neutral substitutes to linear polysaccharide chain, that is hydroxypropyl solubility, viscosity, stability, solution. Like this polysaccharide can be modified. Hydroxyethyl or hydroxymethyl can be incorporated. Here it can be used as different vehicle for drug also. Okay. Solubility, viscosity, binding acid group, carboxymethyl sulfate increase solubility viscosity. As a result, different drug prepared with these binders, with these excipients. Okay, they also have to make like different eye drop solution can be made, different nasal preparation, um, ointment, different preparation, pharmaceutical preparation can be made. There are different heteropolysaccharides where different type of monomer units are there, like glucomanans, arabinoxylans. From the name you can see glucose and mannans, then arabinoxylan. So can be linearly branched to varying degree with the different type of branch points. Typical sugar units found in the polysaccharide are T-galacturic acid, T-mannose, T-mannose, T-ribose, L-rhamnose, L-arabinose. These are the important heteropolysaccharides. Okay, so heteropolysaccharide example in the starting. Now, homopolysaccharide offering used in the food stuff like a glucose. Here, one glucose is present. No, sorry, homopolysaccharide example the arabinose xylose. Here, the structure and linear where the package will be there that has been described. Now, when those are these are the which we where it can be found has been described. Now, heteropolysaccharides occurring in the food stuff like arabinose, and these are different compounds which can be found in different plant cell or seeds, brown seeds, levels, and it can be used in different purposes also. Now you can get different type of carbohydrate slides from different websites where you can get different knowledges. Because it's not a very short chapter, so here it is another. So you here. Just 1 second. Classes of monosaccharide. Well, the same you will finding. Polysaccharides are the structure. You can see monosaccharide, saccharide, trisaccharide, saccharide, and structure here. So this is the linear structure where six carbon is attached and hydroxy and hydroxy groups are alternate. That is simple. The aldehyde and alcohol actually glucose. Chemically you can know as the aldehyde or alkyl. Okay. Alcohol is found. Polyhydroxy alcohol. Aldehyde is also there. Benzaldehyde. Okay, this is the responsible for aromatic bonds and chains. Example, and food having different smell. So that is due to compounds. Ketones. This is biologically important compound. Okay, that is pyruvate, and it is present in different form also. So next, thank you, and so that is also described. Okay. Then different type of carbohydrate proteins, their structure, their linkage, their different alignment, their different glycosidic bond here. So I'm showing this is the different diastereomers. Okay. 600 hybrid protein. This is the glucose unit. You can see the structure. CH2H, HOH, HOH. This is the D This is the alpha anomer and beta anomer. Here you can find the structural alignment. See here, two hydroxy groups are below the level, and this is the cis and trans beta anomer. Structure of pyranose structure. Reducing sugar, there is reducing sugar. Carbohydrate with the free or potentially free aldehyde here, and that can be reduced, that can be reacted with different Benedict solution. Depending on this reduction property, there are different identification taste also. So this is the Benedict solution. CO2 and CO, H2O with CO2. This is forming a glucose. This is forming. This is glucose aldehyde group is oxidized to COOH. Okay. So glycosidic bonds. So that is another part. Whenever two glucose units are joined, one hydroxy group will be attached from another compound and forming a acetal. So this is the hemiacetal. Here hydroxy group is attached and it is forming a acetal compound. OR hydroxy group is in acetal. So these are the different structure you have to study, not so much, and we give you questions important from this chapter. Thank you all.